Hi, my name is Jesse Murray, and this summer I did a virtual internship at NASA Ames Research Center working under the mentorship of Diana Gentry on astrobiology statistics research. Specifically, I worked on a NASA project called SCOBY, which is short for the Statistical Classification of Biosignature Information. The goal of this project is to build machine learning models that classify samples as indicative or non-indicative of life based on chemical, photographic, or other data. These models are important because they provide an independent line of evidence that is based on agnostic and algorithmic methodologies rather than human domain expertise. In an actual life detection mission, the predictions of these models could provide an important second opinion to human judgments, and perhaps more importantly, these models can suggest the distinguishing properties of life and the most informative measurements that would need to be made in a life detection mission. In fact, it was suggested in a 2018 paper, The Ladder of Life Detection, that there is a need to bring together different biosignatures and understand how their combinations affect the overall confidence level of a biogenic or abiogenic classification. The work I did this summer provides a starting point to answer this need. To begin my research, I focused on elemental abundance as a data type because of its wide availability among a diverse range of sample types, which I believed was necessary to have in order to train a generalizable and reliable model. I obtained a number of non-indicative samples, for example, Martian salt or basalt, and a variety of indicative samples, which generally fell into three categories. First category was indicative mixed, which was largely inorganic with some biogenic material, for example, agricultural soil or seawater, and indicative non-alive samples, which were fossils, coal, chalk, crude oil, etc., and indicative alive samples, including humans, bacteria, biofilms, etc. I then standardized all these samples to the same simulated limit of detection of emission, which was 1,500 parts per million. And then I trained and validated 4 classification models: k-nearest neighbors, logistic regression, linear support vector machines, and Gaussian naive Bayes. In order to understand the important features, the important elements of the predictions of these models, as well as their prediction directions, I ran the k-nearest neighbors on the 3 leading principal components of the training data, and I ran the linear regression and linear support vector machines with L1 and L2 regularization. The models were validated on 40 to 60 trained to validation ratios, which is rather conservative. The models found that, in general, carbon, calcium, and chlorine were widely agreed upon indicators of life. Additionally, nitrogen, potassium, and phosphorus were also indicators of life. For predicting a sample as non-indicative of life, the models found that silicon, magnesium, aluminum, and titanium were indicators of non-life. Additionally, some elements had varied predictions. For example, iron, hydrogen, oxygen varied widely, and sodium, manganese, and sulfur were varied elements. The performance of these models was rather good and ranged around 82 to 94% for mean accuracy scores and area under the curve for the receiver operating characteristics. These scores are generally good considering that the models were trained on just 40% of the data and then validated on the remaining 60%. In general, the sensitivity was greater than the specificity, which is arguably the trade-off you would hope for in a life detection mission. In future work, we hope to expand the data types to obtain even more predictive combinations of features. Namely, we hope to include isotope fractionation And also, we'd eventually like to include nonlinear models, for example, neural networks. I'd like to thank my mentor, Diana Gentry, as well as Averis, a previous intern, for his work in early data collection. Thank you for listening.